In these lectures, we will be focusing on cell anatomy of both prokaryotic and eukaryotic cells. A number of these anatomical features we're going to return to repeatedly throughout the term because they might be targets for antibiotics, or they might be important virulence factors for pathogens, or which are related to a pathogen's ability to cause disease. So the first couple of lecture videos are going to be on prokaryotes with a focus on bacteria. So we'll talk about the anatomy and physiology of prokaryotic cells. We'll talk about antibiotic considerations. We'll talk about how they move, locomotion, and we'll talk a little bit about how they reproduce. I'll cover archaea a little bit. So again, as I've mentioned, archaea are not the focus of this course because they tend not to be human symbionts. But we'll talk a little bit about their similarities and differences between archaea and bacteria, and also archaea and eukaryotes. And then finally, we'll talk about eukaryotes. So when it comes to the eukaryote section, a lot of the anatomy you can just get out of your textbook. I will provide it in the lecture videos, but you can decide whether you prefer to get the anatomy from the lecture videos or whether you prefer to get it from the textbook. It's just going to be a list of all the organelles and their functions. But then we'll also talk about the different types of eukaryotes there are. We'll talk about medical considerations with eukaryotic infections, or infections with eukaryotic pathogens, and we'll talk about the evolution of eukaryotes. All right, so let's start with prokaryotes. Again, prokaryotes include two domains of life, bacteria and archaea. We're going to focus on bacteria since archaea are not known to cause disease. Bacterial species can be differentiated in a lab by their shape, their chemical composition, what they require in terms of nutrients, what kinds of molecules they use as an energy source, and most accurately by their genetics. And I'm going to leave most of the discussion on laboratory identification to your micro lab, since that's pretty much all you do in micro lab is learn all the different ways that we have available to us to identify what microbe you're looking at in a petri dish. So this being the lecture, we'll talk a little bit more about the lifestyles of these microbes, how they cause disease, those kinds of considerations. Bacteria and other prokaryotic cells divide through binary fission. This is clonal reproduction, and it's really simple. First, the DNA is copied, so you have two identical chromosomes, and then the cell divides around these two different sets of genetic material. And so this is clonal reproduction. Aside from random mutations, binary fission produces two daughter cells that are identical to each other and that are identical to the original parent cell. So now we'll talk about bacterial anatomy. And a lot of these anatomical features should be familiar to you from your prereqs. I'm going to sort of breeze through the material that you should have covered prior to this course and let you review them in more detail in your own time from your textbook. So the review pieces of anatomy are going to be the cytoplasm, DNA, ribosome, cytoskeleton, and cell membrane. You should have at least a vague idea of what these are. So I'll just give a very brief review of them. So the cytoplasm is the liquid inside of the cell, and it's 80% water. But because of the other things dissolved in it, it tends to be quite viscous. So viscous means sort of like syrupy. And so the kinds of things that you'd find dissolved in the cytoplasm are proteins and sugars and electrolytes, things that the cell needs to live. DNA. So DNA is the genetic material. All living things use DNA as their genetic material. So bacteria and archaea have a single circular chromosome, and then also tiny little circular plasmids. Now this is different from our cells, which have many chromosomes and they're linear. So they're disconnected from each other. They're like a straight line. So any eukaryotic cell has at least two chromosomes because there's sexual reproduction, which means you have a chromosome from each parent. So at least one chromosome from each parent. And different species have different numbers, but it's going to be at least two for eukaryotic cells. So for prokaryotes, though, there's a single circular chromosome. So it loops back on itself, and there's just one. And then there are these tiny little loops of DNA called plasmids. So The plasmids are not considered to be a part of the bacterial DNA because they're sort of like rogue genetic elements, and we'll get to them later in the course when we cover bacterial genetics. But they, first of all, they're not necessary for life. The bacteria doesn't need the plasmids to live. They do provide genes that affect how the bacteria behave and the things that they can do, the stresses they can tolerate. But you can think of the plasmids, the single circular chromosome has all of the genes necessary for life, while the plasmids provide the bacteria with sort of like superpowers. Like maybe it provides the bacteria with antibiotic resistance, or resistance against heavy metals, or tolerance for extraordinarily high temperatures. So these sort of like extra superpower genes are going to be on the plasmids. They're not required for life, but they certainly affect how well the bacteria survives. And they also replicate independently from the rest of the bacteria. So that's what I mean by them being rogue genetic elements. They're sort of responsible, they are responsible for their own replication and for their own transfer to other bacteria. So they can move from one bacteria to another, sometimes across bacterial species. So plasmids need to be inside a bacteria to replicate, but they are not completely under the control of the bacteria. So that's why I call them rogue genetic elements. And when we talk about viruses and bacterial genetics, we'll see other examples, different kinds of rogue, DNA and RNA tend to be sort of weird, and sometimes they act independently from a living organism. Ribosomes. So the job of the ribosome is to read the genetic material and build a protein from it. So DNA is blueprint for protein. So the cell membrane, which is made out of lipids, various styles of vitamins, some of our hormones that are lipids, those are not encoded in the DNA. DNA codes for proteins, and proteins build all of those other kinds of molecules in the cell. Okay. So DNA codes for protein, and I like to think of proteins as the little machines that get everything done in the cell. So proteins can be enzymes that build other molecules or break other molecules down. Enzymes conduct chemical reactions. Proteins can be defense molecules like antibodies or other antimicrobial compounds. Proteins can be communication molecules like hormones and neurotransmitters. Proteins can be portals that allow things into and out of the cell. They can be structural. So proteins are the little machines that get everything done in the cell, that do all the cell maintenance. So DNA codes for protein, protein builds everything else. Now ribosomes are the protein building factories. So the ribosomes read the DNA. Well, they don't read it directly, but we'll get to that later. But ribosomes read the genetic material, and they use the genetic instructions to build the protein. All right. Cytoskeleton. So the cytoskeleton to the cell is the same as your skeleton is to you. It maintains the structure. It's structural support for the cell. And the cytoskeleton is made out of protein. And then cell membrane, and I have that in red because even though it's review, the cell membrane is the barrier that divides the inside from the outside of the cell. So it keeps the internal environment of the cell separate from the external environment. And it's made out of phospholipids, but I'm going to, in the next lecture, I am going to spend a lot of time on the cell membrane, even though it's review, because I really want you to understand how the chemistry of phospholipids are responsible for their role in the cell, because how does the chemistry of a phospholipid allow the phospholipid bilayer or the cell membrane to form spontaneously without the direction of any living force? And how does the chemistry of phospholipids provide the cell membrane with its role as a semi-permeable membrane that allows some substances to cross but not others? So we are going to spend a lot of time on that, because when we talk about the chemistry of the cell membrane, we'll also talk about the chemistry of water, which is really important to understand when you're getting down to the microbial level. All right, so here is a little bit of anatomy that may not be review. So you have probably learned that prokaryotes do not have organelles, but there is an exception. They have what we call inclusions or granules. Inclusions and granules that's just two different words for the same thing. These are membrane bound. So that means that there's a phospholipid bilayer, a membrane that contains these inclusions, and they're storage areas. And different inclusions or granules might store different things, like nutrients. So on the slide I have phosphate, carbohydrates, lipids, sulfur. So these different kinds of nutrients can be stored in these granules. They also can store water and things like that. So these are organelles. They're not typically discussed when we talk. We say that prokaryotes don't have organelles and eukaryotes do. This is the exception. All right, so in this diagram you can see, so the cell membrane is sometimes called the plasma membrane, so that's labeled in red here. That's this yellow layer. And then inside that, just the clear space, that's the cytoplasm, the liquid that everything is floating in. And then in purple, you have the single circular chromosome. It's it's very, very long, just like our DNA is very long. So it's all it's like yarn that's all crumpled up. So you can't see here that it's that it's circular, that it loops back on itself. But if you were to stretch this out, it would loop around on itself. So it's one big loop. So that's a single circular chromosome that prokaryotes have. And then there's a plasmid here. So that's that little rogue genetic element that's responsible for its own replication and transfer to other bacteria. And then in blue, you see these little inclusions. So on my last slide, the inclusions were quite large, but in this diagram, the inclusions are very small, so different species will have different kinds of inclusions. And then these little red dots that are floating around everywhere, those are the ribosomes, the protein building factories. Now, in the next slides, I'm going to talk about the various layers outside the cell membrane. So we'll get into the chemistry of the cell membrane, then we'll talk about the layers outside the cell membrane, and then we'll talk about all these little appendages that the bacteria have. And so a lot of these features are going to be brand new for you.